Well, this is the third session here, and we really want to talk about how do you write the methods section of a research protocol. And one of the analogies I used kind of up front was that the protocol in general is kind of like a recipe for how to do the study. And in fact, the true instructions, not just the overview of the dish, but the true instructions for what you're making come down to the methods. And that's what we're going to go through in some detail. Just as a reminder, if any of you want to download these free research templates that we have for different study designs that you can actually use to help populate your method section, and in fact, the approach that I'm talking about is just based on one of these protocols, you can download them for free here. And then the kind of live writing word plugin that I was just talking about, you can download that here, or at least be put on the wait list to get first access to it for free and to test it out and provide some feedback if that's of interest. Our hope is to make a AI writing assistant that actually understands clinical research and best practices and can help bake that into the actual writing tools that you're using, as opposed to using something like ChatGPT, where who knows what approach it's going to be taking for a lot of this stuff. And so, again, if methods are, or if a protocol is a recipe, then the methods are really the recipe book. And the purpose of the method section in a research protocol is to really allow anyone else to replicate the study, so that if somebody took the same population, the same inclusion criteria, did the same study procedures, they should be able to replicate your study with enough detail that doesn't leave any kind of unanswered questions. And I would say that one of the biggest problems I see with research studies that people are designing for the first time is that the methods aren't detailed enough, and it leaves kind of ambiguity that when you actually start the study, you have to figure these things out on the fly, and they might not always be, you might have committed to something that forces you down a pathway that you might not want to actually commit to. And so it's worth saying that there's a lot of different study types out there. So these are the actual study types involving kind of patients. So you could have observational studies, or you could have experimental studies. But then there's also meta-research studies like systematic reviews and meta-analyses. And regardless of the study type that you're actually performing, there's probably an optimal protocol and structure for the method section. And so if you go to the templates that I shared earlier, there's different kind of protocol templates for different study types. For example, trials versus systematic reviews versus observational studies. But if you wanted to get more specific, each of these probably has a specific subset of things that you should specifically consider. And this is not, in our kind of 30, 45 minutes together, this is not going to cover a comprehensive graduate degree versus of research methodology. It's just impossible. But hopefully I can give you a little bit of a flavor for some of this. And I'll just be checking the chat every once in a while as we go here, just to see what the questions are. Perfect. All right. So for the sake of this protocol that we're going to discuss today, I'm just going to choose an observational study, because most of the kind of early studies that we do as kind of more junior researchers are observational in nature, because they're simpler, they don't involve interventions, they're less complex from a regulatory perspective and an ethics perspective. However, many of the trials that you might participate in as a research assistant or research associate might be more interventional, because many of the trials that pharmaceutical or medical device companies are doing are interventional by nature, but not all.
Mm-hmm.
And so I just want to throw out something from the get-go that I think is probably the biggest take-home for designing a research study. And that's that you don't have to start from scratch. Again, the templates that we have, or go to Equator Network. And Equator Network is an organization designed to improve research quality. And they've come up with these things called reporting guidelines. And what reporting guidelines are, they're checklists for things that you should be considering when you're designing, and also when you're writing out your research study at the end when you go to publish it, so that other researchers and other readers can understand the research that you've done. And for each study design, there's a different reporting guideline. So for randomized controlled trials, CONSORT; for observational, STROBE; systematic reviews, PRISMA. And these checklists have about 30 items. This is only about half of it. And they really go through section by section. And they say, all right, study design, present the key elements of study design early in the paper. The setting, so describe exactly these things in your setting. This is how you should describe your your participants based on cohort, case control, or cross-sectional. These are the variables you should consider, and it goes all the way through. If you follow this as a blueprint for most study designs, it actually gets you pretty far, and it'll ensure that you're not failing to consider important aspects. And this is really what all the protocols that I generated, the templates were based off, were the best practice reporting guidelines for those individual study types. So I'll just pause here and see whether anyone else has any questions. Nope. Excellent. So let's cover a couple specific sections that an observational study should include in the methods section. Now, this might be slightly different than a trial, like an interventional trial. This might be different than a systematic review. Again, individual subsections for those are available in either in Equator Network or in the protocol templates that I have. But this is how I would think about it for an observational study. So let me share the protocol here. Let me share my screen, exit out of that, pull this over here. Excellent. All right. So this is just kind of what the methods of a protocol look like for most of my studies. So I start off with the ethics approval, a broad statement about kind of study design, which we'll go through, the reporting guideline, kind of adherence, saying which reporting guideline we use to help generate this, the setting. We're going to go through this in a bit more detail, but you can see it's quite specific. Which hospital, how big is the hospital, what services are available at the hospital potentially. Inclusion criteria, which probably actually should be called eligibility criteria here, because eligibility criteria includes both inclusion and exclusion criteria. How patients are going to be recruited, sample size calculations, the actual study procedures, how they're going to be recorded. And you can see that there's a fair bit of detail here for how things are going to be recorded so that it's very, very explicit. Talking about what thresholds are going to be used in the study, how we're actually going to collect data, including like exactly what data are we collecting, age, sex, mods, comorbidities, and all of that. Then we go into primary outcomes, secondary outcomes, descriptors of the cohort, statistical analysis plan with how we're going to deal with missing data. And we're going to go through this in more detail, but I just wanted to show you what it kind of looks like when it's all together. You'll have your main heading of methods, and then you have these different subheadings along the way that will help shape the discussion. So stop sharing that for a second. And we will start sharing this again. Can we give the slides? Potentially, we can give the slides afterwards. Yeah, we could share them afterwards. Sounds good. All right. So at a minimum, you need to report the name of the ethics board that will be obtaining approval for your study. You should describe the consent process, and then as appendices, provide the consent forms or letters of information, or LOI/consent form. And the consent process really varies depending on what study design we're talking about. So for an interventional study, by definition, virtually all of them require consent from research participants, like informed consent form being signed by the patient or a... My microphone has been changed. Let me change back here. I don't know what happened there.
We can hear you well.
I do. 1 second. Right there. All right. But by definition, so interventional trials need informed consent. Observational studies, sometimes you can get waivers of informed consent. So for example, for retrospective research, you're not going to approach people retrospectively for consent. And even for some prospective observational studies, if the observation is low risk, if there's no kind of harm to patients, you might be able to have a waiver of informed consent, but that's going to be up to your local ethics board. And there's some situations where it is appropriate or not appropriate to do so. And that's typically guided by regulatory bodies surrounding ethics based on your country. And so in Canada, we have the Tri-Council policy, the TCPS2 policy, and there's similar policies in most countries that ethics boards adhere to. The next thing is the reporting guideline adherence. And we already talked a little bit about this, but I like to be explicit in the protocol which reporting guideline are we using, and then actually use it. And this is something funny. We did a research study a few years ago where we looked at studies that talked about reporting guidelines. So the study would say, hey, we use the Strobe reporting guideline. And then we actually looked to see whether they used the guideline. And even though people say they use it, they were slightly better than the people who said that didn't say they used it, but they weren't adhering to it very closely. And so if you are actually using a reporting guideline, please actually use it because it is important and it just improves the quality of your manuscript. And it shouldn't be an afterthought. And that's one of the challenges with reporting guidelines is that people think that they're just when you're finishing your paper, oh, you just use it to put the final polish on. But I would really encourage you to take these to the heart of the research because these are really important things that allow that impact bias and allow adequate interpretation of your future research. REB, great question, Condal. Research Ethics Board would be the term REB. All right. So the next thing is a section on research design. And typically I like to keep this a very high level, brief description of the study, such that somebody, when they first go into your methods section, they know, is this a diagnostic accuracy study, a cohort study, an interventional study, a systematic review? And so it typically reads one or two sentences like we will conduct a prospective cohort study in the ICU to evaluate the association between this disease and this outcome. Or perhaps it would be we conducted a pilot or a phase 2 randomized control trial in the emergency department to evaluate the effectiveness of drug X compared to drug Y. Or we can we will perform a systematic review and meta-analysis of patients admitted to the emergency department with lower back pain to evaluate the effectiveness of this management strategy versus this management strategy. So it really just from the get-go defines what the broad study type is that is going to be performed, and then you go through that in much more detail in the coming sections. The setting. So really readers need to know where your study took place and understand the context of that environment so that they can understand whether or not your research is applicable to their setting. So if I'm practicing in Canada and I saw we have attendees in Nigeria and Dominican Republic, if we were talking about the intensive care unit and that we have versus in some places elsewhere in the world, their resources and staffing might be very, very different. And so findings that are generated from research in one setting may not be generalizable to other settings with different resources or practice patterns. And so that's why it's important to say, all right, that they're going to be recruited at which hospital or hospitals. Is it like an academic or community hospital? What city and country? What dates are they going to be recruited from roughly? And what services that are relevant to that disease are available? So if you're doing an oncology study, talking about what oncological services are available at that center might be relevant. For example, is there stem cell transplant? Is there advanced CAR T therapy? Similarly, if you're doing a cardiovascular study, is there a cath lab capability? Is there a CCU? What sort of subspecialty cardiology services are available? This may impact the interpretation of the results of the study. Just looking at some of the comments here, I see a question. Yeah, so what if our setting later extends to other locations also after the protocol is written, like a satellite site? So great question. I would say though that you should know whether or not your protocol is, whether you plan to extend to satellite sites before you start the protocol. Because most research studies, if you are going to be expanding, they don't just expand like, oh, it went really well at one center, we're now just going to start adding more centers. Typically that's pre-planned out by the investigator team because like you need to know which centers you're going to be recruiting at from a funding perspective. Do you have enough money to actually bring those centers on? But then also from a sample size perspective to understand what is your expected recruitment rate. So I would say that if you add more centers down the road, you should actually specify that in the protocol. Which centers are you going to add? When are you going to add them? Why are you going to add them? And that should be predefined. Now, there might be rare situations where a center wants to come on board and you're like, oh yeah, that sounds great. In fact, we did that with a study where a center approached us saying, hey, we really want to start recruiting patients for this. Can you add us? Absolutely. You can always update the protocol, but it's just, you don't want to make it the habit that you're just kind of adding people in without consideration, because adding another center really impacts things like data analysis. For example, are you going to stratify your analysis based on the centers? How are you going to look to ensure that the whatever intervention is being applied consistently between centers. There's a lot of consideration, so it should be done with a lot of forethought, and it's not something that you just kind of throw out there. Great question. All right, so setting, and then we go to participants. And so this should really include how patients or data sets, if they're not papers, were identified and recruited, but don't report statistics of it. And this is something I sometimes see in this setting where people actually put some data surrounding the participants, like their age and sex. Save that for the results. I mean, we're talking about protocols, not manuscripts, so this isn't as applicable, but just something in general. And so things that you want to include are the eligibility criteria, which are the inclusion and exclusion criteria. Pretty straightforward. You want it to be very clear which patients are you going to recruit and which patients are you not going to recruit. And I would say that one thing I see is sometimes people, when they submit protocols to me the first time, they have like one or two inclusion criteria. Almost certainly when you start asking more and more questions, that inclusion criteria and exclusion criteria list goes up to more and more and more. And so if you ever have an inclusion criteria of just one or two things, chances are you probably haven't thought about all the reasons you should be excluding patients. Now, a good example in our intensive care unit is, and this is something that people always forget, is like if you have a patient who's going to die imminently, you don't want them recruited in almost any study, because unless the study's surrounding death. And the reason why is that no matter what intervention you choose, if somebody's like almost on death's door and they're kind of beyond hope, applying any drug or intervention will be unable to benefit them because they're already too far in that disease process. They're already too close to death. And so, for example, thinking about how are you going to deal with patients that come in that meet all the criteria, but like there's no chance of them surviving, you should probably exclude those criteria. And how do you exclude those? Or similarly, if you have patients with advanced organ failure, cancer, metastatic cancer, because the more and more of these, what I'd call prognostic factors people have that portend a really bad outcome, the less likely most drugs or interventions are going to modify their disease course. So I just throw that out there. There's a question here. Could exclusion criteria be thought of like the niche we're performing the research on, so most statistics could apply there? Yeah, I would say that eligibility criteria are really trying to focus down the population such that it is very, very clear and reproducible, so that you're not including patients that have a lot of different disease processes. It's about narrowing the population and making it as homogeneous as possible. Because if you study almost any drug or intervention in an extremely heterogeneous population, where some patients have different severities of disease, some patients might not even have the disease, some people are earlier in their disease course, later in their disease course, more severe, mild, more comorbidities, more treatment, less treatment. If you combine those all into a study, chances are whatever drug or intervention you're studying will not show a difference, because there's so many other factors that we haven't considered. So that's why most clinical trials, you're trying to recruit as homogeneous of a population as you can, such that the only differences between patients that get treated and not treated are the intervention and not random chance due to comorbidities, for example. Yeah. The next thing I do want to talk about a little bit is sampling, because this is something that people totally overlook, but for observational research and interventional research is probably one of the most important things that you can do right or wrong. And so, again, without this turning into like a master's epidemiology course, there's really two types of sampling. There's probability sampling and non-probability sampling. So non-probability sampling essentially means that different individuals in the population you're going after actually have different chances of being chosen. And a good example of this is, say you're looking, say you want to use a new test. So I do ultrasound research. So say you want to do ultrasound and you want to scan people that come into the emergency department. But ultrasound is difficult on patients that have higher body mass index, that are obese, for example. So you're waiting in the emergency department to recruit patients, and you see somebody come to the door and they're really obese, and you're like, ooh, I'm not going to choose that guy. Like, I know it's going to be tough from the get-go. Let me wait for somebody who has a lower body mass index to come in that I'm going to get better views on. immediately you've done what we would call convenience sampling. So you're selecting people when you have a research associate or a researcher available to recruit the patient, or you're using some sort of kind of like researcher biased way of selecting people. And the problem with that for observational studies is it can lead to selective sampling due to factors that the researchers may be implicitly biased towards. So it might not even be that a researcher is trying to like skew the results, but it could easily happen. And so really you want to either do random sampling, or in observational studies, one of the best ways to do it is you do what's called consecutive sampling. So you just say, we're going to take every patient that comes in with that meets this 24/7 or during the day, so that there's no chance that we're going to say, oh, we're going to take you, but not the next person. So consecutive sampling is better than convenient sampling. And then if you have a massive amount of patients you could sample, then something like random sampling might be more appropriate. But you can see that sampling itself has a lot of nuance and there's a lot of different approaches towards it. All right. So the next thing that it's important to talk about is data collection. So what data was or will be collected? I wrote this in the past tense, but it should be future tense for a protocol, past tense for a manuscript. So what data will be collected? Um, you want to be explicit, and this was where I think in the first session I talked about I actually create shell data tables for like the demographics, exactly what demographic data are we collecting, exactly what outcome data, exactly what observational data we're doing, such that it's predefined from the beginning what we need to collect. Who's going to collect that data? Because that might influence things. So if you have an outcome, for example, that is subjective, like let's say the outcome that you're collecting is days that patients are confused in the intensive care unit. If it's a trained neurologist or ICU doc collecting that data, they might see the chart and they might be like, oh, I think the patient was confused here. Whereas a research assistant without medical background might miss that. And so who's collecting the data is actually very important. And then when did they collect the data? Did they collect the data live, like the day that things happened, or did they go back a couple months later, or will they go back a couple months later to collect the data? How did they collect the data? So was it using paper charts, electronic medical records, patient surveys? That's very relevant. And then what did the data collectors know? And this is something crucial when you're actually designing your study. You don't want your data collectors that are collecting objective data to know the outcome of the patient, which arm the patient was assigned to, because theoretically that could bias data collection. And now you're going to say, isn't data collection totally objective? Like how could it be biased? It's just like yes or no. But what you'll find is actually when you're collecting and recording data, there's more subjectivity in it than what you might think. So for example, let's say you're trying to collect a blood value that on day 7 you want to collect a hemoglobin value to measure somebody's red blood cells and how much oxygen carrying capacity there is in the red blood cells. Great. But imagine if on day 7 they have two values done. Which one do you choose? They're done 1 hour apart. One slightly higher, one slightly lower. You have to choose one of them. You could average them. You could choose the first one, the second one. You're going to have to make some sort of subjective decision on that. But if you know that the patient died, or if you know the patient survived, it might implicitly bias you towards choosing one versus another. So this is just something to be mindful of. So I'll pause here. Um, yeah, just looking here. So what is the best way to prevent researcher bias sampling while creating the research protocol? What section here do we go into the detail of exclusion and inclusion section? So that would be under participants that you go into exclusion and inclusion criteria. And then in terms of sampling and how to prevent bias in sampling. So I would say that in general, avoid convenient sampling. Convenient sampling will always introduce the highest amount of bias because it's researcher driven which patients you're going to pick. If you're doing observational research, consecutive sampling is really, really good. Or just in general, consecutive sampling is good. But there's a limitation. Can you actually recruit every single patient that comes into the hospital for 24/7 or a clinic 24/7? Probably not. Your research team very likely does not have the infrastructure to actually support that. So then what you can do is you can instead say, all right, we're going to consecutively sample within this window. And so that could be from 8 a.m. to 5 p.m. when there's a research assistant available, Monday to Friday. That could be 1 day a week that you consecutively sample every Monday. But even that can introduce some bias. And this is interesting. And it was something I never thought about. Because let's say you're looking at a study for surgical patients, but every Monday they do the most complicated surgical cases because Dr. So-and-so, the world-renowned surgeon, he operates on Monday. So all the most complicated patients happen on Monday. You might actually get a biased representation of outcomes based on the fact that on certain days of the week, patient acuity is different in the hospital. And we actually see that in our intensive care unit. So for example, in our intensive care unit, the most complicated and sickest patients undergoing cardiac surgery, elective cardiac surgery, often happen on the Thursday or Friday, because then they have the whole weekend to recover, because we expect them to recover a little bit more. So in these situations, you might want to consecutively sample like Monday 1 week, Tuesday the next week, Wednesday the next week, Thursday the next week, Friday the next week. So you could actually change the day of the week that you're consecutively sampling patients. That would be one example. All right, outcomes. So you have to choose a primary outcome. You get one of them. Realistically, you get one of them. Try not to be the researcher that's collecting like 5 or 10 primary outcomes, because then why did you call it a primary outcome? Really, there should be one primary outcome for the study, and then secondary outcomes are kind of hypothesis generating, and be explicit about them upfront. What secondary outcomes and why do you think that these secondary outcomes are appropriate? Now, I'll also make a comment about composite outcomes, okay? So a composite outcome is something like the chance of dying or having a heart attack. So that's your primary outcome. You're like, I can't choose between it. I can't choose whether or not we care more about people dying or more than people having a heart attack. So why don't I just say patients who either die or have a heart attack? And now, if you're familiar with drug trials for like cardiovascular research, this is super common. They will often have composite outcomes of stroke, heart attack, or death, or stroke, heart attack, heart failure, or worsening heart failure symptoms, or elevated troponin and death. And you start getting these really, really big composite outcomes. And the reason why people use composite outcomes are that they increase the number of events or the number of patients who experience that outcome. So maybe only 1% of patients die, but maybe 3% of patients die and have a heart attack, and maybe 5% of patients die, have a heart attack, and have a stroke, and maybe 10% of patients die, have a heart attack, have a stroke, or have a heart failure hospitalization. The more events that you have in a study, the less patients you have to recruit in your study. And so the whole reason to choose composite outcomes is really feasibility of doing the research. But there's a problem with it. And the reason why is not all items in the composite outcome are treated equally in terms of how important they are to the patient. So if I was a patient, for example, and the composite outcome was an elevation in one of my blood markers, having a stroke or dying, I care a lot more about having a stroke or dying than I do about having a little bit of elevation in one of my blood markers. So that would be a bad composite outcome. So a general rule when you're making a composite outcome is that the different parts of the composite should be of equal patient importance and roughly equal prevalence. So you should have the same number of events for people who die, who have a stroke, and who have a heart attack. And patients should roughly care the same between all of those in terms of how important they are to the patient. Otherwise, it's probably a bad composite outcome. The second note that I wanted to make on kind of feasibility studies is that often when we start research, we want an outcome like, all right, I want to show that this drug or this intervention is going to reduce people's chance of dying, reduce mortality. That's great. Chances are, no matter what study you do, you're not going to be able to show that. There are very few trials in clinical medicine that have ever shown this, and these are massive trials that take years and years and years to do with millions of dollars of research. When you first start a research study, I would highly encourage you to choose feasibility or a feasibility metric as your primary outcome, or at least consider it. Because we start a lot of big research studies that are doomed to fail before we ever started. A good example is you start a big trial, but then you realize that the recruitment rate was way slower than you anticipated, and the consent rate was lower. and actually the adherence to the protocol was much lower, and the amount of drug given to the patient for various factors was much lower. Those are all things that could be caught in an earlier study that just focuses on, hey, is this big study we have dreamed up even feasible? Well, instead of spending $10 million and doing the study over the next 5 years, only to find out that, oh shit, like our protocol sucked, why don't you do a small study for 3 to 6 months where you recruit 20 patients? And the outcomes you're looking at aren't patient outcomes like mortality, but process outcomes. How quickly are you consenting patients? How quickly are you recruiting patients? And you also learn a lot about the how to do the study in those first 3 to 6 months that you can then apply to the broader kind of study that eventually you might dream of. So I'll pause there for a second as well. All right, any questions about this? Okay. Statistical methods. So again, this is not a master's in epidemiology. You need to talk about your methods. You should say, what software are you using to perform your analysis in? How are you going to handle continuous variables, not continuous variables, categorical variables? What sort of p-value threshold is considered significant, and any other methods that you have applied. And related to that, if you are choosing to do some sort of multivariable analysis, how were variables selected and what models kind of were built and why. And this might be getting a little bit more advanced, but I just want to make a little comment about multivariable analysis just because this is now best research practice. Previously, maybe like 15 years ago, there was a trend to, when you're doing like multivariable analysis and regression, to first doing univariate. And if all of this just goes right over your head and you're like, I have no idea what he's saying, you can just turn your brain off for 2 minutes if this isn't of interest to you. That's totally okay. It might be of interest to some people who are more interested in kind of the stat side of things. And this is a relevant change in the field of multivariable analysis in the past decade. So previously, what some researchers would do would be they would look through their univariate analyses and look at the associations and figure out which variables had the highest association, and then they would then take the variables with the highest association and put them in a multivariate analysis. Don't do this. This is essentially been shown to be like flawed for multiple reasons. Just because there's a strong association doesn't mean that they should be included in the multivariable analysis. And then also, you might have variables that are very much collinear, which means that maybe patients develop renal failure and patients with heart failure. It's actually the same process that leads to both of them. So including them both in your analysis might actually cancel things out. Instead, what you should do if you're trying to do a multivariable analysis is identify which variables biologically, based on previous research or your own experience or your research team's experience, might impact the outcome of interest that are not kind of that don't compete with each other. Then you could include those in the analysis, for example. So as opposed to choosing it based on the results of univariate analyses. All right, so if you just tuned out for that and got a coffee, that's totally fine. Welcome back. One of the last points I want to make here is sample size calculation. Every study should have a sample size calculation. Now, you might be like, well, like we just analyzed all the data we had. Well, that's okay. Like I'm not saying every study has to have a good sample size calculation, but you should think about sample size for every study, because if you have a rare outcome and only 40 patients, there's no point really analyzing your data to seeing whether or not there's an association with that outcome, because almost certainly, unless the treatment effect was so massive, you're not going to show a difference. And so being really mindful that the more rare your outcome is, and the more rare your outcome is, and the less of an effect that whatever exposure in an observational study or intervention in an interventional trial has on that effect. So the lower those are, the bigger the sample size you're going to need. And that's just crucial when we think about designing studies, because often our studies are underpowered, and often we choose sample sizes, and this isn't the right way of doing it, but often people choose sample sizes based on what they have funds for to recruit patients, but not based on actually what is most appropriate. I do want to throw a comment here about if you're doing a pilot study and you're doing a sample size. A lot of people think like, oh, well, you can't calculate a sample size for pilot studies. You actually can, and we have a sample size calculator specifically for that down below at resub.app/ps or pilot study sample size. And then if you need sample size calculations for observational research, I highly recommend the riskcalc.org/sample size calculators at the bottom. They're fantastic and they can really help give you an estimate of sample size for these studies. And so I'll pause here for a second. I'll share the protocol and we'll go through the protocol one more time kind of with this new lens. But I'm curious. And any questions, comments? Yeah. I think this is admittedly this is a bit tough because like you can't really teach how to write a methods like in terms of the theory behind the methodology in one session because each of these points could be hour-long webinars or even courses in a graduate study. For example, in my master's in health research methodology, we spent an entire course talking about sample size calculation and multivariable regression. It's a semester of graduate studies just to cover one of these points. And so what I hope this gives you is a bit of an overview for what we should consider. And then, you know what, just like kind of we mentioned from the get-go, like this is something you're gonna have to learn and read about and read papers about and really take it upon yourself to learn how to do this. So hopefully this gives a scaffolding that you can start filling in more advanced information in. All right, so ethics approval. Ethics approval will be obtained through this ethics board. I didn't talk about consent here, but that would be something to do. This is that generic kind of study design statement. So this study is a prospective observational cohort study assessing venous Doppler in patients with septic shock admitted to the ICU. All right, you have a pretty good idea about what study design we're talking about here. Then you go into the reporting guideline stuff. So this is going to be an observational study here. It's going to be at these hospitals, and there's some details provided about, all right, this ICU is this big, and this ICU is about this big, and these are the types of patients that we would expect to find them in those ICUs. We have inclusion criteria, so adult patients who meet 3 or 4 of these criteria within 12 hours of ICU admission, so these kind of things. And then at the time of enrollment, patient with goals of care restrictions or limitations on management will be excluded. Perfect. Recruitment, the sample size calculation, of course. Then we talked about the actual study procedures that are going to be done, which I spent some time describing. Exactly what data is going to be collected down to the final variable. So everything is there, and those would be summarized in a table here. And then these are the clinical variables being collected, and then these are kind of the echo variables. And so every single thing is totally explicit. And what this allows us to do is, like me as kind of the PI of the researcher and the kind of person trying to come up with a study idea, I can now hand this off to a research assistant who can actually take this and generate the case report forms and the data collection forms and actually conduct the research. The primary outcome will be study feasibility outcomes. Now, you know what? I broke my own rule, and technically I chose a few different primary outcomes for the study, but when it's feasibility, like you could say like a couple different feasibility outcomes. So recruitment rate, consent rate, and completion of each part of the protocol were all kind of important feasibility outcomes. And then secondary outcomes include things like mortality and the things that you would traditionally be more interested in. Statistical analysis plan, so how are we actually going to analyze the data, and how are we going to deal with the missing data? So if we're missing something, are we going to try to impute it, which means try to figure out what that value would be based on your best guess? And there's different ways to do imputation, but we decided that we wouldn't do it and we'd just exclude it from the analysis. All right. Yeah, so Claire is saying what is considered an adequate sample size? That's a great question. So it really depends on what your study design is and what your outcome is. There's different calculators, but essentially you want to make sure that you have enough of a sample size that based on how much of an effect size you expect, so effect size being like if you're comparing drug in A and drug B, how much better is drug A than drug B for your primary outcome? And then how common is your primary outcome in both of the groups? those variables will be what determines your sample size because you want to make sure you have enough patients that a positive result is likely a true positive result and a negative result is likely a true negative. And there's kind of a balance that you'll see there. Um, a good question about from Neha Patel about can we calculate sample size for a meta-analysis research? Great question. So typically for meta-analyses, you should include all studies that meet your eligibility criteria without a restriction on sample size. If you are encountering too many studies, which rarely happens, it it is probably a reflection of your inclusion criteria for the meta-analyses being too broad, and you should narrow it down. And that's typically what we see. And again, so whenever you're trying to pool data together, be that in a primary study like a observational study or in a meta-analysis, you want to make sure the population that you're including is as homogeneous as possible. And this is especially important for meta-analyses, where if you have a very broad inclusion criteria, you're going to take studies that are actually quite different. And yeah, you can pull them together, but what you're going to find is that you have high heterogeneity and you can't really make any meaningful interpretations because the studies that you're including are just so different. And so I would say that if you're having too many studies, you should probably narrow down your inclusion criteria. A good example is we just did a systematic review where we found like 100 studies that met our inclusion criteria. And we're like, ooh. And so what we ended up doing was instead of focusing on all of these different metrics on ultrasound, we actually just took one metric and then we found like, we're like, oh wow, that's much narrower. So then we did the same thing with every one of the individual metrics, for example. So that would be my comment on that. All right, let me share my PowerPoint one more time here. And so, in kind of keeping with the theme of this series, I really would like to challenge and encourage you to choose the study protocol template that matches whatever study you've been working on. Or if you haven't started, you're not too late. You haven't missed out on anything. Just dive right into this. Try to complete a methods section that you think would be reasonable to answer the study design. Now, if you don't have anything right now, I want to encourage you to do an observational study that is a cohort study of patients that you might encounter as part of your research based on your practice or where you're doing research or where you want to do research, and pick a condition that they have and try to come up with some sort of observational study that you could imagine doing. They're typically the simplest studies to design. The template that I have is really good for them, and it will allow you to go through the methodology quite simply for this. But probably some background reading is going to be required, especially for some of the analyses, for understanding things. Asking ChatGPT like to help explain things and be like, hey, what are some of the things we should include for an observational study? That would be super, super good, and use ChatGPT a little bit as a mentor to help teach you some more stuff about this. And then look up whatever reporting guideline from equatornetwork.org. If you Google Equator Network, like the equator of the Earth, you will find it. And so if you're doing an observational study, find STROBE, which is for observational studies. If you're doing a controlled trial, choose CONSORT, and just go through the checklist and try to apply as many of those things to your protocol as possible, just to make sure that you've considered everything that you can. And so with that, I certainly welcome any questions. The manuscript templates are available here. If you want to sign up for our AI writing assistant, it's no longer called ReSubWrite as of our submission to the App Store. It's LiveWrite. But if you want to check that out, as soon as it's available, we will send it over and you can check it out for free and whatnot and play around with it.